Hello all, welcome to another edition of your Chem 112 lab kit. I'm reporting to you from here, my laundry room lab, which you may have become very accustomed to at this point. But what we're going to be talking about today is lab number 7, which is the synthesis of aspirin or acetyl salicylic acid. So today's lab is pretty straightforward in terms of setup. It's very contained, very little equipment involved. So what we're really going to focus on today are components of the lab kit, where to put them, how to set up for safety. And in particular, I want to give you some tips today about how to do waste disposal. Because in today's lab, you will be working with some pretty corrosive acids. So I want to talk to you a little bit about the safest way to handle them. So as always, reminder, make sure that you're in your safest workspace. We've talked about that. You want ventilation, you want a sink, you want a clean, wipeable surface. So that could be your bathroom, your laundry room, your kitchen, as we kind of have discussed in the past. But what I want to talk to you about first is what materials you're going to need for this particular lab. Now, here's the really good news is the lab kit is sort of self-contained in that we only have to need really one bag of materials. And that bag is this bag right here, which says the qualitative synthesis of aspirin. So you'll see that there, qualitative synthesis of aspirin. And this has essentially everything you need. And I'm going to unbag this in a minute and show you all the materials that we're going to use. But what I also suggest having on hand that they don't recommend in the manual, but I think is really helpful is if you can, I would grab a white piece of paper and a permanent marker of some kind or a pen. Because like we did with experiment number 8, we are going to label our well plates, or excuse me, experiment number 5, which was the chemical reactions. We're actually going to use this to label our well plates instead of having to write directly on it. Because what you'll see is that the well plate is really small. And then the other thing I'd recommend is this beaker from your equipment bag. I would definitely have this on hand. And what I would do is I would add a little bit of water to it. This is going to be as part of our waste disposal. The other thing that I would grab is I would actually grab your baking soda. We're going to use this in the end to neutralize our acids before we put them into the sink. So in conclusion, the kind of the things that I would have in front of you is your qualitative aspirin lab kit, a white piece of paper and a permanent marker, baking soda, and your container of water for your waste disposal. Now, as always, what I'm going to do is I have my kind of self-contained tray to keep all of my chemicals. So we're going to keep all of our chemicals in this tray and we'll have all the equipment outside of the tray. So just to recap, here's what we'll need. I'll lower this camera a little bit so you can see. So we'll put our baking soda in this tray. We'll put our waste beaker in this tray. And now let's talk about what actually comes inside of this kit chemically that you'll need. So in your brown paper bag, this is going to be your willow bark. So this is actually the bark from the willow tree, which was used by indigenous populations for centuries in Egypt, South America, and China. They would take this willow bark and they would chew it, or they would make a tea out of it to extract salicylic acid. So you'll be doing the same thing. Don't chew it. All right. What you'll be doing is you'll be using ethanol to extract this. So you want your willow bark. We'll put that on our reagent tray. And then what you're going to have is a smaller self-contained bag. And this is going to have your reagents in them. Now, if you open this up, what you will see, and note on this, before you touch these, make sure you're using gloves. Because in case any of these bottles have leaked or there's anything outside of the bottles that we don't want to touch, we want to make sure we have a gloved hand. So what you should have is iron 3 chloride. You'll be able to tell because it's that rust color. So if you can see that. So your iron 3 chloride, we're going to put that on our tray. We also have in here our acetic acid, and it says on the side 3M. So this is a very concentrated, strong or weak acid, but it is concentrated. So make sure you're careful in handling this. It'll have a little bit of a vinegary smell when you open it, but don't panic. It's not bad for you to smell it. It's essentially the same thing as a really strong vinegar. So acetic acid. The third reagent that you'll have in here that you're going to want to use, is our ethanol. So this is the ethanol that we're going to use to actually get out the salicylic acid from our willow bark. So we've got ethanol here. So we'd also put that on our tray. And then the last thing we have in this bag is this little scooper right here. So we're going to need that to transfer some of our willow bark and our chemicals a little later. So I'll go ahead and put that spoon outside of my tray on my sort of equipment table. Now, the other thing that you're going to have inside of this equipment tray is you're going to have some allocated plastic pipettes. So we have 1, 2, 3, 4, 5 of them. So these are going to be specific to this experiment. So you want to have all 5 of those droppers in front of you. The other thing that you're going to have is your well plate. Now, if you look at this well plate, what you'll notice is that the wells are pretty small. So we're going to be working with a small amount of chemicals for each one of these experimental runs. And you can kind of tell it's going to be small amounts because if you look at the size of the bottles of reagents, they're also pretty small. So most of this is going to be done on a small scale. So again, we have our well plate that we're going to use to run our experiments. A couple more things, like I said, almost there. So we also have this little guy of pH paper. So I'll show you what that looks like in just a second. But when you open this up, this pH paper, what you'll see is a couple things. So the first thing is this little table. And if I zoom into the camera, what you can see is that each color has a number above it. So for instance, magenta is a 1, blue, navy blue is a 14. Each color corresponds to a pH. So inside of this, along with that, you will have, if you can get it out of the envelope is always the tricky part, right? There we go. All right, so inside of this envelope, you'll have a stack of pH paper. They start out as orange. And what you do is you dip them into your chemical reagents, and then you hold it up to your pH chart, and whichever color it matches, that will give you the pH of your solution. So you're going to have your pH paper in front of you. And then the other 2 things you're going to have is a bag of salicylic acid. So salicylic acid comes as a powder, and acetyl salicylic acid, which also comes as a powder. And that is what this little spoon is for, so that you can scoop these chemicals into your well plate. Okay. Now, in terms of your plate, your well plate, how do we manage that? So one thing. So I didn't want to bore you with me opening this green wrapper, but inside of it is a glass stir rod. So it looks a little bit like this. And what you do is you use this to stir the chemicals in your wells plate. The reason we like glass is because glass is non-reactive, which means that when you dip it into your chemicals, it's not going to react with any of them. It'll just be there solely to kind of mix it up and agitate it. One thing is be very, very careful when you're opening this, because this is really fragile, this glass stir rod. So what I did is I used scissors and just cut the bubble wrap off, but just be really careful of this guy. It's quite fragile. Okay, so the last thing that I want to talk about, or last 2 things, is number 1, what would be a good way to set up your well plate? So as I mentioned before, when you have your well plate like this, if you look on the actual well plate itself, what you will see, and it's kind of hard for me to capture on this camera for the doc cam, but in clear plastic, it's actually labeled. So it says A, so if I'm holding it like this, so long ways on top, what you'll see is that across the top, it says 1, 2, 3, 4, 5, 6. And then along the side, it goes A, B, C, D. So that's actually labeled on your well plate. But if you have a hard time seeing that, you can do one of 2 things. So one, you could take a Sharpie and you could fill in that letter, make it darker so you can actually see it. The other thing that you can do is, and I've done this before, is I'll take my well plate and I'll set it down on my paper. And then what I'll do is I'll label it on the paper. So 1, 2, 3, 4, 5, 6, A, B, C, D. So I'll label it like so you can see that. There we go. I'm going to make you guys all dizzy, but you can see that I've labeled it as a grid on my white piece of paper. So those are just a couple options in case it's hard for you to actually visualize what should be in your plate. Okay, so last thing we're going to talk about is cleanup and waste disposal. So I want to talk to you a little bit about how we would handle that. Now, what's going to happen is at the end of the experiment, what they advise you to do is to essentially take this well plate with your chemicals in it and dump it directly into the sink. Now, technically you can do that, but I don't advise it. And the reason I don't advise it is because you do have salicylic acid in here, you have ethanol in here, but more importantly, that salicylic acid could be slightly acidic, which is not the best for your drains. So what I recommend doing is your beaker with water that you have. So I would take this and I would take your well plate. And what you're going to do is you're going to take this beaker with water and you're going to set it into your sink. And so here you have your well plate with all your chemicals in it. So imagine this is full. And what you can do is you can take a little bit of water. So if you have a bottle of water, you can do that. You can get another cup of water. It doesn't have to be bottled water. And what I would do is I would hold the well plate over that little waste container and I would pour water over it and dump the chemicals into that beaker. So once the well plate has all the chemicals removed, then you can take this and you can rinse it off normally like you would wash dishes. But now what you're going to do is once you have this waste container, and by the way, this will probably be like a pink color from some of your tests with the iron chloride, but you would take this guy, and then what I would do is I would add a little bit of baking soda to it, just to neutralize any acid that's left. Give it a good swirl. And then what I would do is turn on your sink so that the water is running. And with this neutralized solution that you've added baking soda to, you can go ahead and pour that down the sink while the water is running. And then give your beaker a quick rinse. And then I would let this water run for about 30 seconds to make sure everything is clear of your drain. And then from there, you can go ahead and wash these two like you would normal dishes. So that would be my suggestion to actually contain your waste. Now, once you're done, your pH paper, when it's dirty, can just go directly in the trash. Your pipettes can go directly in the trash. That's all fine. But most importantly, what I would recommend is if you're done with your chemicals and there's still some left in the bottles, make sure you put them back into the bag that you unpackaged them from. So this bag here. So I put them back in the bag, seal it up, and put it back into your original kit. So that way, if you have extras and you store it in your kit, you know that they won't leak and they'll be contained in their actual original bag. Okay. So from there, that's all the safety and setup. Please go ahead and read your background and procedure to get more information on the theory behind the lab, as well as the pre-lab questions will help you with that. And with that, happy experimenting and good luck.